to what I think is our third Zoom webinar since the lockdown began at the Daiwa Foundation. For the first time in about a month, I am in the office, and I'm happy to report it's still standing. So today we're talking about an issue that is absolutely key to getting out of the situation that we find ourselves in, vaccines and how they work. And so we're very lucky to have Dr. Masahiro Ono to talk to us about that today. So this isn't very international as far as Ono Sensei and I are concerned. We're both in London, as it happens, but I know that there are people participating from all over the world. And moving on, I'll give Ono-san a brief introduction. So his expertise is extremely relevant at the moment, as he's an immunologist with expertise in T cells, and he focuses on how T cells control immunity and whether immunity develops for this particular virus is something that's very much a hot topic at the moment. So he will talk about how immunity works and immune memory, and also about the potential for a vaccine. So he specializes in immunology. I won't read out all of these details here, but he currently is at University College London, where he's a senior lecturer, and he also holds a visiting associate professorship at Kumamoto University in Japan. And with that, I'll pass over to you, Ono Sensei.
Hello, thank you. Thank you, Jason, for introduction. Can you hear me? Great. So thank you for your introduction and also for inviting me to this exciting opportunity to present our data. I'm now opening my presentation. So today I would like to discuss with you immunity to COVID and how we could develop vaccines and treatment. So COVID-19 infection is caused by the virus called SARS-CoV-2. Often these two words are just exchangeably used, but both mean COVID-19. The majority of people develop mild symptoms only by COVID-19 infection, which includes cough, fever, sometimes diarrhea, nausea, etc. So this is okay. However, 10 to 15% of infected people may need hospitalization. They may have more serious problems. On the other hand, some people may remain almost asymptomatic. So between individual differences are large in COVID-19 infection. So the same single virus can induce various consequences. Some people remain asymptomatic. The majority of infected people are fine, but some people develop respiratory failure, multi-organ failure, and some, sadly, some may die. So what determines between individual differences in COVID-19 infection? When we think about this, we should think about the roles of the immune system. So here is a typical clinical time course and outcomes. So sorry, I'm just moving a small window to see my slide. So now, once infection is established, for a while, people are completely asymptomatic, but at some point, they start to develop mild symptoms like cough, fever, diarrhea, and many will recover, but some people develop severe diseases by pneumonia, respiratory failure, and strangely multi-organ failure too. Then some may be rescued, but some may be not. We know some risk factors for severe disease, including age, heart disease, hypertension, diabetes, obesity. And this is a big area of research, but we are not going to discuss this too much today because I would like to focus on the immunity. So firstly, the virus needs to establish infection in our body, and they exploit our proteins to establish infection. And particularly a protein called ACE2, which is used by human cells to control blood vessels. And this ACE2 is exploited by the virus. And ACE2 is found in epithelial cells, in nose, lung, intestines, and a little in endothelial cells of the blood vessels. And COVID-19 virus infects human cells that have this ACE2. So because these cells are found in nose, lung, intestines, people develop cough, pneumonia, gastrointestinal symptoms like nausea, diarrhea. So it's quite straightforward. So the virus can be activated by a human protein too, so that they can more easily infect human cells. And once infecting, they give their genomic information inside the human cells in order to replicate themselves to finally break human cells to spread infection. So basically the message is that the infection part is quite simple. The virus infects the cells that have the protein ACE2, and the symptoms can be, the mild symptoms can be explained by this. So please, next slide, please. So however, the immunity is not straightforward. It is a double-edged sword. So firstly, we need to eradicate viruses, and we need to use our immune responses to fight the virus and protect our body. So the responses are important. However, any immune responses inevitably damage tissues as well, our own tissues as well. And often the responses become too much, excessive. Then this may cause organ damage. And if it causes too much, then this may lead to a fatal result. Therefore, the balance is extremely important. Next slide, please. So to induce immune responses, there are such key immune cells for fighting COVID-19 infection. There are two major types of immune cells. So firstly, innate immune cells, such as macrophages. So they work as the sentinels. So they are in the front line. They can recognize the danger that there is something like a virus. However, they can't distinguish the types of viruses, and they can't remember so which one they once encountered with. However, they can tell that danger to another group of immune cells called lymphocytes. And the lymphocytes can specifically recognize the antigens of COVID-19 virus. Sorry, this is a jargon. So antigens are some fine structures of proteins or something similar that can be recognized by lymphocytes. And lymphocytes can remember infection. So this is a very important part. So they are the cells that retain the memory of infection. Therefore, lymphocytes are the targets of vaccines. there are two types of lymphocytes. Firstly, B cells. So they are the cells that produce antibodies. And antibodies are critically important for inactivating viruses, for preventing the spread of viruses. So ultimately, vaccines aim to induce such protective antibodies. And there is another type of lymphocytes called T cells, which are my area. And the T cells have very important roles. So firstly, they help B cells. So without T cells, B cells can't work properly. And also T cells can kill virus-infected cells. And without T cells, we can't eradicate virus. We can't clear the infection. And sorry, there are two types of T cells. Many types of immune cells here, but one type of T cells is called killer T cells or CD8 T cells. So they can kill such virus-infected cells. So they clear the infection by removing any cells with viruses. And another type of T cells is called helper T cells or CD4 T cells. And they are a very unique cells. They are the conductor. So they coordinate immune responses. They help CD8 T cells, killer T cells. They help B cells, and they help macrophages and other innate cells to coordinate immune responses to optimize our responses to specifically target the virus without damaging our tissues or organs. So next slide, please. So there is an immune quartet. So macrophage, two types of T cells and B cells. So they collaborate with each other to fight COVID-19 infection. And as I told you, macrophages are the sentinels, so they can capture the virus and digest the virus and present a part of the virus to T cells, to killer T cells and helper T cells. And then they can initiate their actions, and the helper T cells help killer T cells to mature, and then mature killer T cells can kill virus-infected cells. Helper T cells also help B cells to produce antibodies. Then ultimately, killer T cells will eradicate viruses by clear infection, and B cell produce antibodies to inactivate or neutralize viruses and block the spread of infection. So this way, we can win the battle against the virus. Next slide, please. Yes. And importantly, immune cells are actively manipulated by the virus so that they would rather cause damages to organs. They would rather facilitate the spread of the infection. Next slide, please. And in severe COVID-19 infection, there are three major types of problems. And so firstly, on the left, the immune responses may cause severe pneumonia. So then patients can't breathe. They need ventilators. And secondly, they may have a very serious condition called medical shock due to cytokine storm, which can lead to multi-organ failure. So this is rather a circulation problem. So heart, kidney, and any other organs, including extremities, legs, or arms, may not get enough blood. And we also know that in this cytokine storm, T cells are severely reduced by some reason. The third problem is in heart and the blood vessels. And this part is still not quite well known yet. But recently, you may have heard news, you may have read news about some rare diseases are found in the States and in the UK called something like Kawasaki disease, which is a problem in the artery, the blood vessel in the heart. Some may speculate that the heart itself may have inflammation, and other blood vessels may have inflammation, but we don't know whether this is caused by virus itself or this is a sort of autoimmunity. So there are three major types of reactions in severe COVID-19 patients. So this is caused by immune hyperactivation in the lung or hyperactivation of the systemic immune system or the blood vessels or heart. So next slide, please. And in the lung, what happens is that macrophages get too much activated, too much excited, and then they produce the substance called cytokines, especially IL-6 and IL-1 beta are a lot produced by macrophages. And then these cytokines are something like a messenger to recruit other immune cells. And they together cause damages to the lung. So in here, so the white space is the airway. So there, such immune cells are causing inflammation. Then inflammation induces water in the space. Then patients will be drawn, although they are in the lung. So this is the situation where in COVID-19 patients, the lung starts to stop working. And this condition is called acute respiratory distress syndrome, ARDS. This situation, pathological situation, occurs in other severe infections too. But in COVID-19, we know that quite a few patients develop this condition. And please go to the next slide. And now, some treatments have started to target this mechanism. So IL-1 receptor antagonist will block one of the cytokines, IL-1 beta, anti-IL-6 receptor antibody and anti-IL-6 antibody will stop another cytokine, and JAK inhibitor will stop the activation of the hyperactivation of macrophage. So this way, farmers have started to actively block this process in severe patients, and many are now in clinical trials. And next slide, please. So next, about cytokine storm. So the problem is not just about the lung, but it's systemic. And this is a data from the blood of severe patients. And I'm showing here 3 cytokine data. Again, this is the substance that is produced by immune cells. And I said IL-1 and IL-6 are produced by hyperactivated macrophages, and this another one is also produced by macrophage, IL-8. And you can see that in patients with COVID-19, the patients that required ICU treatment and those just hospitalized, both have a lot of such cytokines in their blood, which means that their macrophages are most likely very much activated, too much activated. And next, please. And even more striking finding is that in severe COVID patients who needed an ICU care, ventilator support, show remarkably reduced T cell numbers. So this is the total T cells, helper T cells, and killer T cells, CD8 T cells. And from the left, it's healthy and non-ICU hospitalized patients and ICU patients. And this is a remarkable change. And we still don't know how this is induced by COVID-19. But it is very clear that when patients develop such severe diseases, they will not have the function of T cells. T cells are paralyzed, which must be a significant problem for them. So next, please. And this way, T cell immunity is paralyzed, actively paralyzed in COVID patients. Next, please. So then how about antibodies? Now we talked about macrophages, so they are exploited by the virus, and the T cells are dysregulated by the virus. And then how about the antibodies? And people speculate that antibodies, even antibodies, may be exploited by the virus. And there are some findings in other coronavirus infections and similar viral infections that some antibodies may rather promote the spread of viruses in our body. So this is called antibody-dependent enhancement of infection. So the idea is that antibodies are meant to neutralize the virus, but so by binding viruses, antibodies are meant to block viruses, but because antibodies are easily taken up by macrophages, so through antibodies, viruses may rather infect macrophages to further spread the infection through immune cells. So this is the idea. We still don't know how much this may occur in COVID patients, but this is one concern that immunologists are thinking. Next, please. So this way, the quartet for us may work in a really terrible manner. So hyperactivated macrophages can initiate cytokine storm, which induces lung dysfunction and circulation problems. T cells may participate in cytokine storm, and their function is paralyzed. And so that CD8 T cells, killer T cells, can't clear the infection. And the B cells may produce antibodies which are not enough functional and may enhance infection. Next, please. So now, you may have herd immunity. And when we talk about when the pandemic ends, either herd immunity is established or an effective vaccine is produced. However, we don't know how the immunity to COVID-19 is established in each individual. Therefore, we talk about herd immunity, but as immunologists, I should say that it's a little still premature to make any decisions based on this. So we think that at least eventually, if we get a good vaccine or anything else to protect ourselves, up to 60% of the population, the pandemic is meant to be stopped. Next, please. And so we now learned how our immune system works, how the immune cells fight against the virus. And when we talk about immunity, the memory, there are two types of memories, immunity. One is B cell immunity, and another is T cell immunity. And could you please click the slide to show? Yes. So now we know what is happening behind the infection. And when we talk about measuring COVID-19 immunity, in reality, we are just measuring the circulating antibodies only, basically. So this is measurable. And any antibody test kit will analyze this part. However, the memory is established not only by B cells, but also by T cells. And there is still not yet enough efficient method to analyze T cell immunity. Next, please. Still, we have some knowledge. We know something about how coronavirus memory, how anti-coronavirus immunity can be established and maintained. And there are some similar coronaviruses, similar to COVID-19, such as common cold coronavirus, SARS coronavirus. And these viruses can induce antibody responses, and the patients will be protected by the antibodies. However, the antibodies will decay, usually within a year. So the left panel shows the decay of antibodies against SARS infection after patients recovered. And the right panel shows the decay of antibodies against a common cold coronavirus. So now we don't know yet about what happens with the COVID-19, but many speculate that antibodies against COVID-19 may decay in a similar manner. We will know the results soon because now more and more reliable antibody test kits are becoming available. And as I said, there is not yet reliable methods for measuring T cell immunity. Next slide, please. So about the vaccine, there are many attempts being taken in the world. Some are in clinical trials. Many approaches use new technologies, because there is no available vaccine for any type of coronavirus, including COVID-19, which means that we need to use a new technology. This is a bit tricky part because when using a new approach, we need to carefully check the safety. And it's estimated that we still need some time, up to 1 or 1.5 years before getting any reliable vaccine. And I will not talk about the very details, but would like to highlight some of the attempts. Next, please. So firstly, I told you that the infection is established when virus binds the protein ACE2 on human cells. And the virus uses their protein called S protein to go into human cells. And many of COVID-19 vaccines target the spike protein, S protein, of the virus to block the infection. And the left panel shows, so S protein is on the surface of SARS-CoV-2, COVID-19 virus. And we know that, as on the left panel, recovered patients will produce neutralizing antibodies against S protein. So they bind specifically to the S protein. And we know that in the presence of such anti-S protein antibody, the virus can't infect human cells through ACE2, as on the right panel. Next, please. So here, there was one big concern about the use of S protein as a target for vaccines. And this study was by SARS, not the COVID-19, but in SARS infection, people aimed to develop vaccine against SARS using the same approach, using S protein. And they developed such a vaccine using a little unique viral vector called MVA. And they immunized the monkeys, 6 monkeys, to see the effect of vaccination. And they, in the middle panel, they checked and confirmed that vaccines successfully introduced neutralizing antibodies against S protein. And in the lower panel, what they found was that paradoxically, the monkeys that received the vaccines against the SARS virus rather developed severe pneumonia. So the immune system rather become hyperactivated in this case. So we were very much worried about what would happen in case of COVID-19. And the next slide, please. But there is a good news about this. So there were the Oxford group conducted an animal experiment in the States and confirmed that using their vaccine, which targets the S protein of COVID-19, successfully protected the animals, monkeys, that were vaccinated. So this is a good relief and good news among recent development, and they are doing phase 1-2 studies. And next, please. So these are the treatments in development, and there are 2 types of treatments. One is to target the virus, antiviral therapy, and another is immunotherapy to modulate immune responses. The first antiviral therapy basically aims to block the activities of the virus. This may be a virus inhibitor, for example, to block the replication of viruses or to block the entry, or we can induce antiviral responses of many cells by giving interferon, which is a protein, human protein, to enhance antiviral responses. And this is expected to target mild to moderate patients. Immunotherapy can be classified into 2 parts. One is to block virus infection using antibodies. Another is to stop hyperactivation of immune cells. And to stop general immune cells may be done by corticosteroid or immunosuppressants. And as we discussed, hyperactivation of macrophages can be potentially stopped by anti-IL-6 antibodies or IL-1 beta antagonists. And there are more therapies coming. Next, please. So the entry block by immunotherapy. So the very simple approach is to get serum from recovered patients to transfer the serum into the people who are suffering from severe disease or who have a high risk to develop severe disease. And we know that some preliminary reports already showed that this approach seems to be safe. And many pharmas are further developing a monoclonal antibody approach, monoclonal antibodies against S protein to be transfused into severe patients and high-risk patients. And this will be even safer because this is not serum, but it's an industrial production. So it's free from other infections. And there are multiple attempts ongoing. So next, please. So this way, anti-COVID-19 therapies should target normalizing the balance of immune responses to enhance antiviral responses and to moderate any other immune responses, especially to control hyperactivities of immune cells. So as you can see, especially in severe patients, this is not a subtle issue. We need to probably stratify patients to identify the most optimized patient for each group of patients. Next, please. So this is the last slide. We are based in Imperial College London, and we are still in the lockdown, but have started COVID-19 research from our perspective, from our field, in our field, T-cell biology. And I would like to thank my lab members and also my funders, including MRC. And I'm also supported by Kakenji in Japan too. So thank you everybody for your attention. And I would be happy to answer any question.
So thank you very much indeed today to Dr. Masahiro Ono for a fantastic presentation, which certainly taught me an awful lot more about the science than I have learned from reading various newspapers recently. And so it's goodbye officially, but we'll carry on chatting for a little while.